Hello class. Last time we have learned about the cyanobacteria. In this lecture, we are going to learn about the archaea. In the late 1970s, a distinctive type of prokaryotic cell was discovered. It soon became clear that they also shared many RNA sequences, and the sequences were different from both those of the domain bacteria and the domain eukarya. These differences were so significant that these organisms now constitute a new taxonomic grouping, the domain archaea. As shown in figure, animals, plants, and fungi are kingdoms in domain eukarya. The domain bacteria includes all of the pathogenic prokaryotes, as well as many of the non-pathogenic prokaryotes found in soil and water. The photoautotrophic prokaryotes are also in this domain. The domain archaea includes prokaryotes that don't have peptidoglycan in their cell walls. They often live in extreme environments and carry out unusual metabolic processes. Archaea and eukaryotes share a number of ribosome RNA sequences that are not found in bacteria, and their protein synthesis and ribosome subunit structures are similar. Other ways that the archaea differ significantly from other cell types are that certain genetic sequences are found only in their ribosome RNA. Their cell walls are also quite different, being composed of polysaccharide or protein and lacking peptidoglycan, and some lack cell walls altogether. Archaea cells, like bacterial cells, exhibit a variety of shapes. As shown in figure, cocci and rods are common. Both usually exist singly, but some cocci form clusters, and some rods form chains. Curved rods, spiral shapes, and pleomorphic archaea have also been observed. As shown in figure, some archaea exhibit unique shapes, such as the branched form and the flat postage stamp shape. Archaea cells also vary in size as much as in shape. Typical rods are 1 to 2 micron wide by 1 to 5 micron long. Cocci are typically 1 to 3 micron in diameter. However, extremely small and extremely large archaea have been identified. Several free-living, acid-loving microbes measure a mere 0.2 to 0.5 microns in diameter. Before they were distinguished as a unique domain of life, archaea species were characterized as being either gram-positive or gram-negative based on their response to gram staining. Thus, like all cells, they will stain either purple or pink when gram-stained. However, their staining reaction does not correlate reliably with a particular cell wall structure as it does for bacteria. Archaea cell walls exhibit considerable variety in terms of their chemical makeup. Furthermore, their cell walls lack peptidoglycan. There are many different types of archaea cell wall. The most common type of archaea cell wall is an S layer, composed of either glycoprotein or protein, as shown in figure. The layer may be as thick as 20 to 40 nanometer. Some carcinogens, salt-loving archaea, and extreme thermophiles have S-layer cell walls. Other archaea have additional layers of material outside the S-layer, as shown in figure. Methanosporellum has a protein sheath external to the S-layer. Another methanogen, methanocyanin, has a polysaccharide layer covering the S-layer. As shown in figure D, in some archaea, the S-layer is the outermost layer and is separated from the plasma membrane by a peptidoglycan-like molecule called cellulomurine. Cellulomurine differs from peptidoglycan in that it has L amino acids instead of D amino acids in its cross-links, N-acetyltylosaminuronic acid instead of N-acetylmalamic acid, and beta-1,3 glycosidic bonds instead of beta-1,4 glycosidic bonds. These differences mean that lysozyme, penicillin, and other chemicals that affect bacterial cell wall structure and synthesis have no effect on archaea cell walls. The last type of archaea cell wall consists of a single, thick, homogeneous layer resembling that in gram-positive bacteria as shown in Figure E. These archaea lack an S layer and often stain gram-positive. Their wall chemistry varies from species to species, but usually consist of complex polysaccharides. Archaea membranes are composed primarily of lipids that differ from bacteria and eukaryotic lipids in two ways. First, they contain hydrocarbons derived from isoprene units 5-carbon bronzed molecules. Second, the hydrocarbons are attached to glycerol by ether links rather than ester links. Ether linkages are more resistant to chemical attack and heat than are ester links. Two major types of archaea lipids have been identified: glycerol diethers and diglycerol tetraethers. Glycerol diether lipids are formed when two hydrocarbons are attached to glycerol. Usually, the hydrocarbon chains in glycerol diethers are 20 carbons in length. Diglycerol tetraether lipids are formed when two glycerol residues are linked by two long hydrocarbons that are 40 carbons in length. Tetraethers are more rigid lipids than diethers. Despite the significant differences in membrane lipids, the basic design of archaea membranes is similar to that of bacteria and eukaryotic membranes. When carbon-20 diethers are used, a typical bilayer membrane is formed. When the membrane is constructed of carbon-40 tetraethers, a monolayer membrane with much more rigidity is formed. Archaea that live in moderately hot environments have membranes containing some regions with monolayers and some with bilayers. Overall, the cytoplasm of archaea cells is very similar to that of bacteria. Within it can be found inclusions, ribosomes, a nucleoid, and plasmids. Cytoskeletal proteins have also been identified. Let's summarize the difference between bacteria and archaea. First, the cell envelopes of bacteria and archaea are one of the distinctive features. Bacterial membranes contain lipids built by attaching fatty acids to glycerol by ester linkages. Archaea membranes link isoprenoid hydrocarbons to glycerol by ether bonds. In some archaea, the membranes are monolayers rather than bilayers. Second, nearly all bacterial cell walls contain peptidoglycan. This molecule is unique to bacteria. Archaea cell walls are much more varied than those of bacteria. Many are composed solely of S layers. Ribosomes of bacteria and archaea are the same size but differ at the molecular level. The rRNA molecules are similar size, but the nucleotide sequences are distinguishable. Furthermore, the ribosomes of at least one archaean have an additional rRNA that is not observed in bacterial ribosomes. A few ribosome proteins are unique to archaea. Archaea also differ from bacteria in their metabolic capabilities and gene expression. Okay, that's all for today's lesson.